Good afternoon. This is AP Biology Immune Video 2. Immune Video 1 was focusing on non-specific immunity, what we now call innate immunity. And this video is going to focus more on specific immunity, what we now call adaptive immunity. The first video is sort of like skin. That's a general barrier for all bacteria and all viruses. Or some of the phils, neutrophils, basophils, eosinophils, phagocytes. Those are types of phagocytes that can hopefully generally just eat whether it's E. coli or strep or staph or whatever. What we're focusing on in this video is a more specific immunity. So for example, I might build up antibodies to the flu of 2017, either because I got it or because I got a vaccine for the flu of 2017. But by the time the birds, generally waterfowl, as they migrate, they're the ones that are carrying around flus, so all flus are kind of bird flus. By the time they make it back in 2018 and then again in 2019 and 2020, every year the flu has mutated as they as they migrate around the globe. And so when that happens, now I might be really good at dealing with the flu of 2017, but by the time it comes back as the flu of 2018, it looks different. So my body was great at killing this exact flu, but if it tweaks a little bit, I'm screwed. Kind of like SARS, MERS, and now SARS-CoV-2 that's causing COVID-19. So we learned last time that a macrophage, we didn't have this MHC II up here last time, so I'll explain that part, but we did learn that a macrophage might gobble up a bacterium, phagocytosis. Once the bacterium is phagocytized, then lysosomes will surround the phagosome. That's what we would normally, what you're used to calling like a vesicle, but if we just sort of engulf this, they call it a phagosome. And so they will surround that. The phagosome is phospholipid bilayer. The lysosomes are phospholipid bilayer, so when they merge, now this guy basically gets chewed up by the digestive enzymes like proteases, for example, in the lysosomes. Sometimes we can find other things in lysosomes. We can find things that are more similar to like hydrogen peroxide and stuff like that. But anyway, we digest this up, and now we're going to present it to the world. So real quick before I get into that part, so that'll be new. This right here is an exploded neutrophil. So neutrophils are kind of interesting because what they'll do is if they roll up on a single bacterium, they'll gobble it up. Phagocytosis, they'll do this. If they roll up on a second or a third or a fourth, they'll keep doing that. But if they essentially find themselves sort of surrounded by bacteria, you're way outnumbered, instead of trying to take out one or two, what this neutrophil will do is he will basically just explode. And as he blows himself up, he's sort of showering everyone around them with chemicals that can destroy them. However, he also releases these things that look like nets. They're called nets. And they're actually made primarily out of DNA, so it might just be more of a natural selective kind of convenience. But as he explodes, his DNA ends up sort of like gluing them all together. It'd be almost like if I went into the middle of a crowd, and as I'm basically blowing up like a set of grenades or something, I know it's really violent, as I blow myself up with a bunch of grenades, I'm also like sending out a net so that as people try to escape and they're getting like covered with maybe I have toxic gases or something, maybe it's not a regular grenade, then now they're like stuck in there. So these orange bacteria are like stuck in the neutrophil nets. And like I said, they're actually called nets, N-E-T-S. Pretty interesting. Okay, we'll leave that picture up there because it's kind of cool. So now, back to this. I just gobbled up this bacterium. However, what's the likelihood if I got a cut and now bacteria are inside of my cut, what's the likelihood that like 5 bacteria got in? Or 100? Or 1,000? Keep in mind, supposedly a cubic millimeter of human tissue is supposed to contain millions of cells, perhaps a billion cells. So bacteria would be more than that. So if I have a cut, then I might have millions of bacteria that got inside. Hopefully not. So this guy right here should probably tell other people. So he'll go in, he'll gobble up the bacteria, phagocytosis. So as he chews this guy up, then what he'll do is he's going to hold out a piece of it for the world to see. So let's say that this bacteria had I know it's not showing up real well on the screen right now. I'm just making these look like little triangles. So these little spikes on the edge of the bacteria are looking like triangles. There's a reason I'm picking triangles. You're like, why would you care? But so he's now basically displaying one of those little pieces of the bacteria, the triangle, in this thing called MHC II, Major Histocompatibility Complex 2, Major Histocompatibility Complex. Histocompatibility is tissue compatibility. The fact that my brother and I have similar MHC proteins, there are many different types, means that if I needed to donate a kidney to my brother or vice versa, we might have a shot. The fact that you and I likely, if we're not related, we likely don't have very well matching MHCs. And that's one of the reasons that your kidney, if you donated it to me or vice versa, our bodies would probably see the incoming kidney as a foreign invader. So that's why they originally called it major histocompatibility, tissue compatibility. It's a major tissue compatibility complex. Okay, so pause that for 1 second. In the immune system, it gets a little bit tricky. So the immune system is a little bit like it's almost like a circle. And wherever you start, and you start explaining something, to fully understand it, you had to understand the moment right before it. So what we do is we just sort of like start somewhere, make it through a whole cycle, and by the time we make it through a whole cycle of how all the cells work together, then it sort of makes sense. Now, obviously, we're not going to have like a normal test over this stuff this year in 2020. If you're watching this video sometime later, then it could be. So for those of y'all that are going to be taking a test on this, maybe in future years or whatever, I always draw these particular cells with these particular structures. If I try to always remember it on the fly, then I end up having to have brain processing power going back and forth. So if I was getting ready to, like for example, whenever I was getting ready to teach this lesson, the way that I know I'm ready for this lesson is I just take a Post-it note and I draw out these 6 cells with all of these various proteins on them. And if I can explain the immune system. If I can't, then I gotta make sure I know my stuff before I get up here in front of you guys. So, pause that and draw those on a piece of paper somewhere. That reference will make it easier. That down there, if you can't quite tell what it says CD4 matches with MHC II. CD8 goes with MHC1. That says MHC1 on self cells. Macrophages have MHC II. NK cells have this thing that's like a little hook. I'll explain that later. TH, that's a helper T cell. It has a CD4 protein and a TCR, a T cell receptor. That Y-shaped thing is a T cell receptor. TC, cytotoxic T cells, that sounds deadly, has a CD8 protein, and it also has a Y-shaped T cell receptor. And then B cells, those contain an MHC II, class 2. This is like technically class 2, class 1, class 2. B cells have a MHC class 2 protein, and they have a B cell receptor that's kind of Y-shaped on them. So we will come back to this chart a lot momentarily. Okay, so I present this to the world. So essentially it's like this. If I'm a macrophage and I gobble this guy up, I'm like, gobble, gobble. Then I break down parts of it, and my MHC II goes from the surface of the cell right here, goes into the phagosome, grabs a piece of what I've been eating, and it basically shows it to the world. So it'd be almost like I grab a piece of hamburger, I chew it a little bit, and then I'm like, here world, here's what I'm eating. And the reason I would do that is to warn everybody, hey, be on the lookout for these orange guys. They look kind of like this. They might have these little like triangle looking spears. Okay, so that is over here happening. Meanwhile, nearby, a helper T cell, TH. Helper T cells have a CD4 protein and a TCR, a T cell receptor. So meanwhile, over here I've got a helper T cell with its T cell receptor and its CD4 protein. Now, helper T cells they mature in the thymus. Remember T cells, thymus, not thyroid, thymus, on top of your heart. And those receptors are made at random. So here's the thing that's kind of weird. Pretend this is the first time for my body to ever encounter this particular bacterium. Or pretend that this is SARS-CoV-2, and it's a novel virus, meaning it's a new virus. My body hasn't seen it before. We don't have any, we don't believe that we have any natural immunity to it, it seems. So let's say that this is a bacterium, or it's a free-floating virus. Once viruses get inside of my cells, remember, viruses enter our cells, hijack them to make them basically virus factories, and that's how they hurt us. So a virus, once it's inside of our cells, we deal with it a different way. But before it gets inside, it's free-floating, just like a bacterium. The way that bacteria typically hurt us is they never enter our cells. We saw an example in the last video of like listeria, tuberculosis also does this, but typically the way that bacteria hurt you is they just hang out in the back of your throat and start secreting something that wrecks the back of your throat, and now you have strep throat. So bacteria are easy in a sense because they look different. So if I'm an immune cell, I can go around and check, that's a Mr. Cotton cell, that's a Mr. Cotton cell, that's a Mr. Cotton cell. Not only is that not a Mr. Cotton cell, that's like a prokaryotic cell. It's a bacteria, so I can attack it. Well, if a virus is free-floating or a bacteria is free-floating, we would deal with them the same way. So this could be the spike protein, that's why I drew it that way, for SARS-CoV-2, for example. Okay, so my helper T cells make receptors at random. So we kind of revealed this a little bit earlier in the year. Let's say that I've never been exposed to this pathogen, never been exposed to this bacterium. I should have a receptor that fits it. The example that I've given before is, let's say that we told you, hey, 50 days from now, a key is going to arrive, and you need to have a lock ready that fits that key. And you're like, can you tell me anything about the key? And it's like, no, we don't even make it yet. We wait until the moment before we send it, we cut the key in 1 minute, then we mail it to you. So what could you do? You have a key coming, you know nothing about it, it doesn't even exist yet. How could you have a lock that fits it? You now basically just make as many random locks as possible, and you hope that one fits. So if I make 1 billion locks at random, I'm probably gonna find one that fits that key. That's how your immune system works. So this might be pretty quick. Macrophages might be eating up bacteria pretty quickly. But to involve your specific immunity are the letter cells like T cells, helper T cells, cytotoxic T cells, B cells. These guys are all specific immunity. That's why they're one color. These guys are not. But again, these are all letters, like that's a T cell, that's a T cell, that's a B cell. So specific immunity takes longer because we have to find a match. So I've just got millions of different helper T cells rolling around with different receptors on them. And I'm just hoping that I have one that matches. So after several days, maybe, I'll actually find a match. That receptor happens to fit that particular bacterial protein. So here's my helper T cell. He has a CD4. You might remember over here, we said that CD4 matches up, or CD4 can talk to MHC II. So CD4 can grab onto this MHC II. So that little brown guy right there is MHC II. And this CD4 protein can latch on. Now from there, once they latch on, we can see if there's a match. We can see if the orange spike protein matches up to this T cell receptor. And the helper T cell tries this, not a match. Again, not a match, not a match, not a match. After a long time, we find a match. Just like whenever they ship the key over, we'll try it, and what's the likelihood it fits the first lock? Probably not. So we try a lock, nope, 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 nope. Maybe like a few days from now, we happen to find, just by luck, the right key and lock combination. That's what's happening here. So in this analogy, the orange is the key and the purple is the lock. We just make them at random. Okay, so now we finally find a match. By chance, maybe there's like, I have no idea. I'm just gonna pick a number, 5. Maybe by chance there's like 5, 6, something like that, of these helper T cells with that exact receptor on them that we just made at random. Again, if I'm making locks at random, I'm probably gonna make a few of them exactly the same, by chance, right? Pick a number between 1 and 1,000. Do that a few thousand times and you're gonna sometimes pick the number 774 more than once. So I'm making these at random. I've got maybe 5 of them. So what happens next is this helper T cell is going to make clones of itself. It's going to make monoclones, individual clones of just this one thing, monoclone. And some of those helper T cells are going to remain around for another day. That says memory if you can't really tell what my M's are. So I make clones. And I'm just going to pick a number. I'm going to say 10 million. I'm just wanting there to be a big gap. 5 versus 10 million. How much money do you have? $5. How much do you have? 10 million. That's a big gap. Big difference. So I make clones. Maybe 5 million are memory cells. And maybe 5 million are what we call effector cells. Those are the ones that basically go to work. So those are the ones that are ready to fight now. Memory cells, they hang out for another day. That way, if I get infected by this bacterium again, the exact same one, instead of taking days for us to find a match, because I've only got 5 locks that match, now I've got 5 million locks that match. So now, when I start trying to see if I have a match, it doesn't take me very long. So now I've got like 5 million effector cells. So here's a helper T cell. And now he is open for business, so to speak. CD4. He has that specific receptor. Now, helper T cells, I kind of compared to the general of the army, and a lot of teachers do, the boss or the general of the army. So the helper T cell kind of like orders the rest of the soldiers around. The helper T cell doesn't do any killing directly. So again, just like a general in an army isn't out there on the front lines typically, they're behind the scenes telling the soldiers what to do. So this helper T cell has to boss around some of the other cells. So now this helper T cell is going to try to get a B cell involved. So this is where it gets slightly odd, so hang with me. All this just happened. Meanwhile, over here nearby, simultaneously, or 4 hours earlier, or whatever, meanwhile, over here, I've got a B cell. My B cells have a B cell receptor and an MHC II. B cell receptor, MHC II. Meanwhile, this B cell happened to run into another one of these guys also. So let's say this is an E. coli. Well, there's more than one. So he runs into this E. coli also. His receptor happens to match. His receptors are made at random, just like T cell receptors are made at random. So there might be, I don't know, half a dozen of these B cells around, maybe 6. Eventually, the right B cell bumps into this particular bacterium. So now that B cell wants to fight. So he's going to make clones. He doesn't have permission to fight yet. He's a great fighter, but he doesn't have permission. He needs permission from his general, the helper T cell. So this guy will make tons of clones. Some will be memory for another day. Maybe 5 million, I don't know. And then some are going to fight right now. Just like before, we call those effector cells. And again, maybe we make 5 million of those. So now, this B cell is great at killing. However, he needs permission from his general. So what he's going to do is he is going to phagocytize this bacterium and display a piece in his MHC II. Then these two are going to talk to each other. Remember, helper T cells have a CD4. CD4 can talk to MHC II. So this B cell is going to eventually bump into this helper T cell with his spike protein that he pulled off of this bacteria. Let's make those triangles on there. This kind of spiky looking protein. And that helper T cell is going to say, yes, that's the same one that I'm after. So the helper T cell is basically saying, look, I got news from macrophages saying this guy is a bad guy. I now get information from you saying he's a bad guy. So now that we've kind of done like a double check step, I'm going to give you permission to go and start killing. If you've ever watched any movies, the way it works in the army, and Navy or military stuff, if you're on like a nuclear sub, so I guess that'd be Navy, according to the movies, you don't just have like one dude over there turning a key to launch some sort of nuclear warhead or pushing one button. No, it's usually two people. There's keys over here. These people have to like punch in the same code at the same time or turn the key at the same time. The reason they do that is so you don't just have some drunk guy over there turning the key, starting a war. Now you have to have at least two people that agree like, yeah, we should start this war. And so that's what we're doing. This B cell is like, I think he's a bad guy. This macrophage is like, I think he's a bad guy. And so the helper T cell is like, yes, I agree, go fight. So you're having like a multiple check situation before these B cells get permission. When a B cell gets permission to fight, then that is when we really start to win the war. This might take 2, 3, 3.5 weeks, but eventually, once this B cell gets permission, that's when it gets interesting. So this B cell has all of these different B cell receptors on its outside. I just drew one originally, but he's actually got many copies. So what he'll do is he's going to become an active cell. You could just call it an active B cell if you want, but an active B cell is called a plasma cell. So this helper T cell gives him permission. He sort of chemically sends across a message, not sort of, he does. He sends across a chemical message and says, yes, you can convert into a plasmid cell. And whenever that plasma cell is active, it just starts spitting out its receptors. So its receptors were stuck in the membrane, they were membrane bound, but now they're free floating. So we just dump them out into the open. So they're now everywhere. Well, remember, these receptors are specific for these bacteria. So now those receptors have a new name. They're not called receptors, they're called antibodies. Antibodies are what we're trying to build up with a vaccine. When we give you a flu vaccine, we're hoping that within about 2 or 3 weeks, you'll build up antibodies. That's why you probably have heard if I get a flu vaccine on January 1st, I probably don't really have immunity until like January 22nd. We'll say about 3 weeks that it takes for this process to happen. If we get flu in you, we're part of a flu. Maybe we just inject this right here. This takes weeks until you're secreting antibodies. Well, now, everywhere that these bacteria are at, so here they are. They were happy. They got their spike proteins sticking out. They're just glycoproteins. I'm stuck on the word spike just because of SARS-CoV-2. SARS coronavirus version 2. And so now these antibodies start sticking to them. So I'll have an antibody that might recognize this, an antibody that recognizes this, an antibody that recognizes these guys right here. And so they'll start like clumping together. Technically, I guess the way that I drew this isn't perfect, but technically one part of that antibody can stick and the other part can stick also. They're bivalent. So you can actually get to where like I can have an antibody and it's sticking to one bacteria here and another bacteria here. They call that agglutination. So for example, if I have blood type A and you have blood type B and you transfuse me with your blood, then now my red blood cells might all clump together. They might agglutinate. So that's what's happening right here. You can't see the antibodies, but the antibodies are what's sticking all those red blood cells together. So that guy and that guy get stuck together in a clump. This guy and this guy get stuck together in a clump. And antibodies are interesting because they're basically a signal that says, kill me. And so antibodies accomplish 3 things. And we'll end up closing out the video with this here in just a few minutes. Whenever bacteria are covered in antibodies, it's basically saying slow repro. It slows down their reproduction. Because of the antibodies being on there, they can't quite divide as quickly as normal. Slow repro, easy phago. It makes phagocytosis easier. In fact, some of the fills are great at eating up bacteria or viruses or whatever as long as they're covered in antibodies. And obviously they can't penetrate cells. The ones that are like listeria and stuff aren't very good at invading cells when they're covered in antibodies. And then the last thing is complement. That's a nice shirt. Now I'm going to shoot you full of holes. Two different versions of complement. Well, with complement, we saw that in video 1. Complement were all those free-floating proteins that go flying around. And what they do is there's actually two versions of complement. We're only going to talk about one. One of the versions of complement works by landing on anything covered in antibodies. So if something's covered with antibodies, that basically says, hey, complement, land here. And so now complement protein number 1 lands on this bacteria, and then 2, then 3, then 4, and 5, and so forth. And then just like we said last video, eventually those complement proteins all form a big pore, and now the cell explodes. Here's the surface of cells covered in complement holes. You don't have to know the name, but if you're in like a physiology class, they call them membrane attack complexes, a MAC hole. Membrane attack complex. And I don't know how well you can see it, but this cell is actually getting ready to explode. It's almost like if I had a picture, if I've got a cell, if I've got a balloon, and just like a regular old air balloon, and I'm gonna poke it with a pen, a P-I-N instead of this, a P-E-N. If I poke it, if you took a picture at just the right time, you would see the balloon as it was like just started to explode, right? With the naked eye, it's just like the balloon's there, pow, now it's all shriveled up in a corner. But if I had a camera fast enough, then the balloon would be like that. That's what you're seeing right here. A cell is exploding. They don't always explode. Sometimes it's just matter rushes in, diffusion, things rush out, osmosis if water's moving in and out, and the cell dies. But they can actually explode. Opsonization. Pretty interesting. So here's the other part that's interesting, and we're in our next video going to talk about vaccines and more specifically SARS-CoV-2 and HIV also, because we've studied HIV a lot. We know how it works pretty well. SARS-CoV-2 is fairly new. So the important thing is macrophages eat, but then they do antigen presentation with MHC. The job of MHC is antigen presentation. It's like, hey, somebody come take a look. Helper T cells say, that's an MHC II, I've got CD4, so we can talk. Yes, I agree, we match. Therefore, I make clones of myself. Now, meanwhile, over here, a B cell eats this bacteria, takes a piece of it, holds it out for the world to see. In his MHC II, he's presenting an antigen presentation. He gets permission from this helper T cell. He's like, I want to kill this guy. The helper T cell's like, I agree, you should kill this guy, go. He becomes a plasma cell, starts secreting all of his receptors, antibodies. When bacteria are covered with antibodies, that will mean slow repro, easy phago, and complement. Now, in the next video, or I'm sorry, let me back up just a little bit. These antibodies, there are different types. If you're ever reading a book and they talk about IgG or something, immunoglobulins are antibodies, and there are different classes of them. Some stay around for a few months. Some stay around for years, decades or more. So the idea is, with me, I got chickenpox back in the day. Now it's a virus, not a bacteria, but it's a free-floating virus, and before it got into my cells, this happened to it. Now I did get into some of my cells. I got chickenpox, but some of them were caught. And I did this, and I made antibodies. Turns out, those antibodies are still floating around in my cells. My memory cells, my memory B cells, I might not have 5 million, but I've got enough. So even though I'm still exposed to chickenpox, I'm 44, and I had chickenpox when I was, before I got into school age. So I think I was like 2, 3, maybe 4. And so for 40 years, I've been immune because I have antibodies that are circulating around that can still remember what it looks like, because chickenpox evolves very, very slowly. I still have memory cells. So that memory cells are what gives you kind of the longer term immunity. However, for certain reasons, and it's somewhat unknown, for certain reasons, whenever we're making antibodies, sometimes you make this one type of antibody and you're immune for a few months. Other times, you're immune for a long time. Some vaccinations, they're good for 6, 8, 10 years. and then you have to get a booster. We have to kind of basically hit you again because your antibody levels in your memory cells were kind of cratering like this, so we give you a booster. So tetanus, you don't get it once. About every 10 years, you're supposed to get a tetanus shot as a booster to keep your immunity up because the antibodies are going down. Let's see, based on time, that's a good stopping point. We're probably about 35 minutes in, I would guess. We'll come back and we're going to talk about cytotoxic T cells, NK cells, and then what happens when a virus actually gets inside of one of my cells. This is easy to figure out who to eat and kill when everything's on the outside. What about when there's a Mr. Cotton cell, and then there's a Mr. Cotton cell that has viruses growing inside, right? So a virus gets inside of me and starts becoming a virus factory, and the next thing you know, I'm just barfing out viruses everywhere. So how do you know the difference between a healthy Mr. Cotton cell and one infected with viruses? And the one infected with viruses, we need to kill that cell. So that'll be our next video. Thanks.